This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 828, recorded on November 11th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from New York, Daniel Griffin.
Hello, everyone.
Daniel, you think you'll ever come back to the incubator and do a live show again?
You know, I was going to do one, last week, but then you were spending time with your wife.
It's a very nice dinner.
Yes. I wonder, maybe either next week, the week after, but yeah, no, definitely. I actually like recording there.
Yeah, it's a nice place. So you're welcome anytime.
Okay. Anytime you're not prioritizing your wife and family above me, which
it's very rare.
Actually, I will share this, maybe our listeners. So I don't know if any of our listeners ever watched the TV series or if you've watched it, Stargate Atlantis.
I have not.
But there's a scene. It's sort of getting near the end of the final season, and there's the brilliant scientist. He's playing you, Vincent, and he goes to a dinner, and who's there but Bill Nye and Neil deGrasse Tyson. And it reminded me of the time that you stood me up and Dixon for a TWiP so you could go hang out with those two.
We never did that.
I think it was. Weren't you going to head up to the what is it where they do the Star Show stuff, the planetarium, and Neil deGrasse Tyson was going to be there, and you okay, you're denying it now. Okay.
No, we never did it. We never stood you up. It's all good.
Because I remember mumbling to my son, and my PhD is not an honorary PhD; it's a real one.
Who has an honorary PhD?
Bill Nye, the science guy.
Okay.
All right. So let's get going. Let me start with my quotation. He who studies medicine without books sails an uncharted sea, but he who studies medicine without patience does not go to sea at all. And that's by William Osler. And I'm going to plan to throw in a few sea stories as we go forward. And hopefully that's a lot of what the clinical updates here do. We get really great deep dives into the science and the literature and the books. And hopefully I get to share some of the sea stories about what we're seeing out there on the high seas of COVID.
Daniel, wasn't William Osler the founder of modern medicine?
A lot of people consider him the founder of modern medicine. Yeah, he a lot of the things that we do, the bedside teaching rounds, the idea that you can learn all of medicine from really a single patient if you just spend the time. It has to be probably a lot of time, but no, they really a lot of the modern ideas. But really, modern medicine, right, is not sitting at the bedside learning from an eminent physician. It's actually looking for evidence-based randomized control studies that really guide us forward. So I guess I'm going to start. You can tell me if I'm spending too much time on this first part, but I went back to listen to our original TWiV special, Coronavirus, SARS-CoV-2 and COVID-19 update, and that was March 4th.
Wow.
I think we recorded it on Monday, and then it got released on Wednesday. But it really just sort of made me wonder, like it actually seemed like we were going to be okay. We were off to a decent start. And I went through that episode, we talked about droplet transmission, but we added how if you were in a closed room with poor ventilation and N95 was recommended. We talked about masks, how they can be, great source control, someone coughing, sneezing. We talked about how for 90% of folks, we expected this to be an outpatient disease, but then 10% would end up requiring that higher level in the hospital. We talked about how critical testing would be because this was so similar symptoms-wise to respiratory pathogens, allergies, really the importance of testing. Talked about incubation time, the time to average onset of symptoms. We talked about telehealth and how we could limit exposures but still manage patients. And we even, this what I thought was sort of telling, we talked about vaccination and how potentially one day we could have antivirals targeting the polymerase and the protease.
Wow.
And then we even went on, like just a few weeks later in episode 606 to talk about how critical proper evidence-based medicine was going to be to guide us going forward.
Wow.
And here we are, almost 2 years later.
Incredible.
So, all right, let's get right into children COVID. Children at risk for COVID, and wearing a mask is less traumatic for a child than being hospitalized. So here I'm going to throw in the first verse of a Robert Frost poem. Two roads diverged in a yellow wood, and sorry I could not travel both, and be one traveler long I stood, and looked down one as far as I could to where it bent in the undergrowth. And my point with this poem is really the choice now that vaccines are accessible to children down to 5 years of age. And it really is a choice to vaccinate or not to vaccinate. You really have this sort of two options in front of you. And not to vaccinate is an active choice, as Ben Franklin learned when he lost his son. Choosing not to vaccinate can often be the riskier choice, the road that puts a child at higher risk. So by the time that our listeners listen to this podcast, over 1 million children in this 5 to 11 years age group will have been vaccinated. And the really nice thing is that we're not hearing any safety concerns. This is going very smoothly. So I think we're getting, reinforced here that the safer of the two choices, the choice not to vaccinate is the unsafe. The choice to vaccinate is a safer choice. And right now, I have to say, the numbers of COVID cases had gone down. They're starting to go up already. They're starting to climb, and unless there's some change in the way this virus and human behavior plays out in the next couple of months, this may be as good as it gets. We're expecting numbers to rise after the holidays. So think about that when you're making these choices. The sooner you protect your child, the sooner you start heading down that less risky path. All right. Testing. This actually lately has been really coming up. And what I'm going to say here is it's really critical. And we've been talking about this on national calls. And actually, I was on a call yesterday talking about it with all our urgent care centers. And I'll be talking about again next week with some of the other urgent care centers from one of the other groups in the area about how critical a lack of resulting delays is going to be when these oral antiviral pills become available. We still need to wait for the exact wording of those EUAs. And part of why I mention this is when those EUAs are written, I am certainly hoping there's no requirement that confirmation be done with a PCR, that this can be much like the monoclonals. If you confirm with a rapid test and we can get people on these pills, we think that timing is really going to be critical. So a lot of the discussions have been with the places where the testing will be. Get ready. We want to not have resulting delays. You don't want to call someone 4 days later to say, oh, you're now outside the window and the efficacy of this oral medicine has been lost because of problems on our end with getting those results. So we are anticipating at the end of this month, sort of right after Thanksgiving, the post-Thanksgiving surge, that we may have the first of the oral antivirals available. And as we get into December, a second may be added to this. So think a lot about, our testing. This is sort of, as I pointed out, this started off as sort of a education update for a lot of our clinicians. And this is something we've got to be thinking about. Time is critical. So if a patient calls, if you're a patient, you want to get that test done quickly. You want to get a result. So that then you can potentially take advantage of these oral antivirals, which seem to really work particularly well during that 3 to 5 days after onset of symptoms. I want to share a story here because I always hear the other stories. So this is one of my C stories. And this was a young woman actually got involved in the case last night. And so the story was this young woman started to feel poorly Thursday night. I'm going to give you the timeline. Started to feel poorly Thursday night. So, being a considerate citizen, considerate coworker, she decided, I'm going to go get one of these PCRs. They have these vans around New York City. You can go, you can rapidly get a PCR test. PCR test came back negative. So she thought, okay, well, must be some other one of your viruses there, Vincent, because you own them all. But and so she figured, okay, well, my PCR is negative. I may be feeling crummy, but PCRs are really good. But weekend went by, got into next week. said, you know what, I'm still not feeling well. And my boss is actually now out from work. So why don't I get one of these and get these tests at home now? So she went ahead, did the test, and it was positive.
Antigen test?
The antigen test. So she thought, you know what, I'm going to get another test. She went ahead and got another antigen test. It was also positive. So sort of as the story goes, right? We always hear the story of, I did the antigen test and it was negative, and then a couple of days later I got a PCR and it was positive. Oh, those damn antigen tests! But just the reality, no test is 100% sensitive. This is a vaccinated woman. She ended up getting COVID. And she'll hopefully ride this out. Hopefully, it will be nothing more than this mild sort of viral syndrome. She'll finish her 10 days of isolation for the infected, and then hopefully get on with her life. But just remember, every test has a less than 100% sensitivity, and timing matters. And really, testing frequency is a critical thing. If the test doesn't make sense, I think this woman made a smart choice. Go get another test.
So this young lady was vaccinated?
She was vaccinated.
So what do you think about 10-day isolation for vaccinated infected people? Does that make sense?
I would like it to change. I think we're going to need the science to make this, but we're starting to get the science, right? We talked about the Singapore study. We talked about the other study. It does look like individuals who have been vaccinated are clearing that viral RNA quicker, about twice as quick as the unvaccinated. So it would make sense as we go forward to start looking at shortening isolation for the infected for vaccinated people. A lot of that, my gold standard for me is really contact tracing. If we can show that we're not seeing transmission past 5 days of symptom onset in the vaccinated, maybe we can shorten that isolation, which would make a lot of sense. Because you know what, Vincent, at some point, predicting the future, you and I are going to end up testing positive, right? I mean, this is here to stay. It's in the deer, it's in the cats, it's in the dogs. We're going to talk about dogs in a second. But yeah, so do we keep isolating for 10 days every time someone gets a positive test? There's a certain point we've got to make more rational decisions. Okay. Active vaccination. Never miss an opportunity to vaccinate. Vaccination is how this pandemic ends. Vaccines, the jabs that keep you from getting sick. I've turned up my autofocus so I can move a little more, and not have that distracting. But here I'm going to tell a hybrid immunity story, right? Because we've been hearing from the labs, people get COVID, they get vaccinated, they have this uber immunity, they're protected, they'll never get any coronavirus, any sort of Becco coronavirus, and so just that's not what we're seeing out here on the high seas. And so I just got a call from, actually it was from the upset, concerned father. And this was a young woman in her 30s. She had COVID initially and actually had quite a difficult time with the initial bout. After a period of time, went on to get fully vaccinated. But then again, last week, started to not feel well. Went ahead and had a positive COVID test. They met criteria, went ahead and got monoclonals, but they had a little bit of a tough time here with this. So, just this sort of reality that, we talk about hybrid immunity, we talk about a lot of serology studies, but, even what we talked about last week, people who get COVID can get reinfected. They can significantly reduce their risk of getting reinfected and getting symptomatic COVID with vaccination, but it's not zero. It's a reduction, but it's not bringing it down to zero. All right, so let's hit a study. The article, SARS-CoV-2 vaccine protection and deaths among U.S. veterans during 2021. And this was published in Science. And I think I've shared my first real job, quote unquote, after I finished my training, actually started during my training, was working at the VA up in Montana. So, thank you for all these individuals for their service. But part of what I liked about this article was they make a really clear distinction, and they even introduced some new TLAs, 3-letter acronyms. They introduced VEI, vaccine efficacy, vaccine effectiveness against infection, and VED, vaccine effectiveness against death. And whenever I see vaccine effectiveness, I always want to see what's that third letter. Is it infection? Is it positive PCR? Is it symptomatic COVID? Is it hospitalization or death? So I thought this was nice that in this publication where they looked at 780,225 individuals in the VA system, they reported that vaccine effectiveness against infection, so VEI, declined, but vaccine effectiveness against death, VED, actually looked like it was holding pretty strong. And so here they talk about for people over 65, they were reporting a VED of 73% for Janssen, 81.5 for Moderna, 84.3 for Pfizer BioNTech. So it's a little bit of a win for Pfizer BioNTech here. We don't see a lot of those. Now, one of the things I did want to make a comment about here, it sort of fits in, is, as I may have mentioned before, I am not seeing much long COVID in vaccinated people who get infected, right? We had that one study out there where we heard about a 50% reduction, but that seemed not even as much of a reduction as I'm seeing. So we reached out, right, to all our different infectious disease physicians at the different care delivery organizations throughout the U.S. My friend Poe out there on the West Coast, Michael Amala, and a few others, and I'm going to leave them out and get upset that they weren't mentioned. John Hitt was on the call as well. Really asking, are you seeing much long COVID in vaccinated people that get infected? And it really was silence. We don't have any in our post-COVID recovery clinic here in New York. This is, to me, really reassuring because that's one of the things that I sort of worried about. I worry about death. I worry about hospitalization. I worry about people feeling ill, the morbidity. But I have a particular soft spot for people that get long COVID and for months and months have lost their quality of life. So sharing that C story.
That's the numbers here, Daniel. How many patients you're talking about?
We're actually talking about millions across the country. So we
Wow.
here, just our care delivery organization in New York, we have over 2,000 providers and we have millions of patients. You go out across the country. So it's actually United Healthcare covers over 100 million lives. And on this call, our care delivery organizations across the country. So this is, I'll say, a large collection of anecdotes. And I look forward to more data on these numbers. All right, passive vaccination. Remember those monoclonals after high risk exposures in high risk individuals. I just had a woman that we took advantage of this today, a woman renal transplant, had a positive COVID PCR, but it didn't make sense. We repeated it. It was negative. We repeated it again. It was still negative. But unfortunately, because of that positive test, they were put in a situation where they were exposed to patients actively with COVID. So remember, think of those monoclonals for post-exposure prophylaxis before you let them go on to get COVID. And I've mentioned that Regeneron has this part of their EUA, but the Eli Lilly product, the bamlanivimab, atezolizumab, has this as well. And here, this I thought was really positive. This is a press release, and this is follow-up data on some information that we saw before. So on November 8th, the 8-month follow-up data was shared via a press release. New phase 3 analysis shows that a single dose of REGEN-COV, this is the casirivimab and the imdevimab, provides long-term protection against COVID-19. So this is the 8-month follow-up data of the study that we read about in the New England Journal of Medicine, the article Subcutaneous REGEN-COV Antibody Combination to Prevent COVID-19, right? So this is getting a under the skin, so subcutaneous injection of the REGEN-COV antibody cocktail. And we've talked about how this only has a half-life of 22 to 26 days. So we were really thinking you're going to have to keep giving these folks this injection every month. But here was just follow up for 8 months. So during this extended time period, there was an 81.6% reduced risk of developing COVID-19 during this follow-up period. So just to give you a sense, how are they catching folks with SARS-CoV-2? They did weekly testing for the first 4 weeks, and then they followed for anyone with symptoms. And if they had symptoms, they would then be tested for COVID-19, for the SARS-CoV-2. And so 81.6% reduction all the way out to 8 months. So, pretty encouraging. All right. Next, moving on to the period of detectable viral replication, the viral symptom phase, right? This is often where the clinical syndrome comes to light. I'd say the time for monitoring and monoclonals, not the time for antibiotics, not the time for steroids. We can do harm with both of those. So, but maybe we're about to get into the time for effective oral antivirals. So I'm going to discuss the Pfizer product and jump in if you want to here, Vincent. On Friday, November 11th, via a press release, Pfizer announced its investigational novel COVID-19 oral antiviral candidate, Paxlovid, that it significantly reduced hospitalization and death based on an interim analysis of the phase 2/3 EPIC-HR trial. So these are, where do those letters come from? Evaluation of protease inhibition for COVID-19 in high-risk patients. There's actually a lot in that title. So this is a randomized double-blind study of non-hospitalized adult patients with COVID-19 who are at high risk of progressing to severe illness. So think of that 10% that we've learned about from our monoclonals. These are people who are either above a certain age, above a certain BMI, have certain comorbidities. The scheduled interim analysis showed an 89% reduction in risk of COVID-19 related hospitalization or death from any cause compared to placebo in patients treated within 3 days of symptom onset. That was the primary endpoint. 0.8% of the patients who received Paxlovid were hospitalized through day 28 following randomization. So that was 3 out of 389. Hospitalized, no deaths, compared to 7% of patients who received placebo. That was 27 out of 385 hospitalized, with 7 subsequent deaths. Similar reductions were observed in patients treated within 5 days. That was not the primary endpoint, but we saw 1% of patients who received Paxlovid were hospitalized through day 28. That was within the first 5 days, so 6 of 607 hospitalized, no deaths, compared again to 6.7% of patients who received placebo, 41 of 612, with 10 deaths in that group. So, all right. Any thoughts there, Vincent?
So the efficacy is a little better than the molnupiravir reported by Merck, which was 50%, I believe, right?
Actually, it's quite a bit better. Yeah, it's quite a bit better. It's when the Merck drug, when Thor's hammer, molnupiravir, was when that data became available, there was a whole discussion like, okay, this is great, it's more accessible, but those monoclonals are still winning. Well, this is 90%. This is actually really, what is a better drug? Potentially, I want both, right? But no, this is pretty impressive. 89%, 90%. This is really getting up there. And the whole idea is this may roll out a week or two after the Merck drug. We're thinking December. Pfizer really communicates well with the FDA, and so the data is being analyzed theoretically as we speak. So, but the key here with the rollout of these drugs is going to be getting those results back in time, right? You don't want a PCR that takes 6 days to get back, and by then you're outside this 3 or 5 day window.
Are they going to be able to make enough doses, both the Pfizer and Merck, to take care of people?
So eventually, yes. The question is how many doses will be available, let's say this December. A few months from now, these are small molecules. This could be pumped out, in large quantity. This is going to be something you take twice a day for 5 days. But yeah, this is what the drug companies do well. This is a lot easier than making these vaccines. So it'll probably be, shortage initially. There'll probably be some restrictions in the EUA. But no, if these get approved, this is something that, massive quantities can be produced. So very encouraging, I have to say. This is, we keep saying this is truly a game changer. This is up there with vaccines. It's not a substitute for vaccines. We still want to get people vaccinated. But boy, this is just another great tool to have. All right. And I am going to jump to the no one is safe until everyone is safe. And here I'm going to throw in another quotation. And this is a quotation from MLK, Martin Luther King. I have quoted this fine gentleman before. On March 25th, 1966, in Chicago at a press conference before his speech at the second convention of the Medical Committee for Human Rights, King said, "We are concerned about the constant use of federal funds to support this most notorious expression of segregation. Of all the forms of inequity, injustice in health is the most shocking and the most inhuman because it often results in physical death." So at this point, I want to tell people, take a moment. First, I want to thank you, but take a moment, go to parasiteswithoutborders.com, click donate, continue to help us get education to the people and places that need it the most. Hopefully we're making a difference, but we're only able to make this difference thanks to you. We're starting to do better here in the United States, but we need to do much better throughout the world. Throughout the months of November, December, and January, all donations made to PWB are going to be doubled up to get $40,000 to support Microbe TV with all this great science education that Vincent and the team are doing.
All right. Time for some questions for Daniel. You can send yours to daniel@microbe.tv. Susan writes, I have a patient with inflammatory bowel disease who is eligible for the third Moderna booster. She's on Remicade every 8 weeks. When is the best time to give the booster?
Yeah, so just for our listeners, so Remicade is an immunomodulatory drug. So it's an immunosuppressive drug. It's targeting the TNF alpha. Now, Remicade is a monoclonal antibody, and we say it has a half-life of about 8 to 10 days. So as you get to that 8 weeks out, you're getting to a very low level, and then you're going to redose it, right? So you're starting with a high and it's coming all the way down. So most of us have been suggesting that people get their vaccinations about 2 weeks before the next TNF alpha inhibition dose. So 6 weeks after the dose, 2 weeks before. Some people are getting this every 4 weeks, so you're going to do it right sort of in the middle there. But yeah, that's usually the recommendation, about 2 weeks before. We don't have any great data looking specifically, so we're really, this is sort of an educated bit of advice based upon the kinetics, based upon the mechanism of this immune suppression.
Chris writes, hello, Vincent and Daniel. As a 56-year-old recipient of the single-shot J&J back in March, I'm curious, conflicted about the need for a booster. After listening to this week's Q&A with A&V, I found myself conflicted even more. I'm only an engineer, but I deal with data and statistical analysis every day, and typically the better data wins the argument or at least moves the conversation in a better direction. My confusion is this. Recently, I've been hearing on TWiV or weekly clinical updates or Q&As how the single-shot J&J isn't good enough, to quote Vincent in last night's Q&A with A&V. However, the article, Durable Humoral and Cellular Immune Responses 8 Months After Ad26 CoV-2S Vaccination, attached, published in New England Journal of Medicine, September 2nd, 2021, which I believe was discussed on at least one TWiV, suggests that the durability of the vaccine is very good. This leads this engineer to the conclusion that in an effort to get us through a pandemic, and assuming the goal remains to be preventing hospitalization and death, both I fully support, by the way, the single dose J&J meets the goal with flying colors 8 months out. Please correct me if I'm wrong, but bring data. I'm all ears.
Okay. Well, this is great. And so here's the way I'm going to put it. We're not talking, or I'll say I'm not talking about J&J as a second dose because of waning immunity. It's really a lack of getting to that high level that we got with the mRNA. So I don't know how much of these data sort of are circulating in the media and the sort of common press, but when J&J was studying their vaccine, they studied it as a single dose, but they also had several investigations throughout the world looking at it as a 2-dose series. And so that's really, I think, what we're talking about with J&J. It gets caught up in the whole booster discussion that we're talking about with the mRNA vaccines. So this was really what is going to give us higher efficacy, J&J as a single dose vaccine or J&J as a 2-dose vaccine series. Now, the 1-dose vaccine actually does quite well above that 50% bar that was suggested was required. When you give it as a second dose, and we've talked about this, they're giving us a 94%, but remember, nice wide confidence intervals that the FDA sort of made some comments about. So that's really what we're talking about with the J&J is we do think a 2-dose series is going to give us higher efficacy than a 1-dose series, but it's not necessarily about waning, actually. And if you look at the T cell response, you look at the antibodies over time, I actually am quite reassured by the J&J data.
So, quoting from the J&J press release, this is the phase 3 ensemble trial, which I think you covered here. 75% overall efficacy against severe critical COVID across all age cohorts in all countries in the study. So that's after one dose.
That's after, yeah, which is
And then
excellent, actually.
with the second dose, it goes up to 100%. So I think that's why you have said in the past, it's a good idea to get a booster. Those are the data, right?
Yeah, I worry about calling it a booster. I really think it's, take your J&J as a 2-dose vaccine series. Yeah, the language around boosters and everything, I think, is more confusing than helpful.
And Chris says, P.S. Don't let my Racanielling fool you. All the podcasts in the Twix family are my all-time favorite ways to spend my commute time. Okay. We don't mind Racanielling as long as it's calm and based on science, right?
Yes, be nice. As long as you're nice, it's great to have having questions. That's what science is all about.
Ingrid writes, my dermatologist prescribed tacrolimus cream for a rash. Could the immune-suppressing features interfere with my COVID vaccine or make it more likely I will contract COVID?
So I do not think so. Now, tacrolimus mainly is going to work on your T cells, but it's not going to really get to systemic levels. So, I think you're going to be fine there.
All right. And our last one from Holly writes, I've searched for an answer to a question about COVID boosters for a specific kind of patient, have not found an answer. I suspect you might be able to provide such information. I have a couple, ages 84 and 90, both vaccinated in February with Pfizer. In September, they were each diagnosed with COVID-19. Neither experienced severe symptoms. They were able to isolate at home. Because of shortages, Denver, Colorado, neither received monoclonals. Now we face the question of boosters. While they both qualify, is there any reason to consider their September infections as a type of booster on its own? Any reason in folks of such advanced age not to proceed with a booster if they should receive boosters? Any advice on the best timing?
Yeah. So this is challenging, actually. And I think we've talked about, the studies where someone gets, say, infection-induced immunity. And then they go ahead and get 2 shots of either an mRNA or the 1 shot of a J&J. And then we look at, for instance, serology or maybe T cell studies. And that actually looks quite impressive. But then I think I shared here a young woman in her 30s, healthy, athletic, who got infected, got vaccinated, still got infected, right? We're seeing that in the studies. We're seeing this out there in clinical practice. We don't have as much data on people that got vaccinated, got infection. Is that a boost? Is that equivalent to getting a booster shot? We certainly don't know. So I think the reason you're saying you're coming up not finding much direction here is that there is not a lot of direction here. So if these individuals tolerated the vaccines well, and you're looking at boosting them, I don't think that infection in the interim is a contraindication in any way. It's certainly not something we're seeing a problem. If they had a difficult time with those vaccinations and now you're sort of on the fence, then again, this is something maybe you want to think through a little bit. Has that helped them in some way? The way I sort of put this together in my own mind, and I think I mentioned this earlier, I think at some point we're all going to get exposed to SARS-CoV-2. It is here to stay. As long as we have a number of years ahead of us, there will be a point. And really, it's going to be a question of how many vaccinations are under our belt before we have that exposure. But yeah, just sort of add a little bit more confusion to the confusion there. There's no clear answer.
If they were your patients, they got no bad response to the vaccine. Should they get a booster after that infection? Would you give it to them?
I wouldn't see that the infection would change how I would proceed. Yeah.
Okay. That's COVID-19 clinical update number 88 with Dr. Daniel Griffin. Thank you, Daniel.
Oh, thank you. And everyone, be safe.